So today we are discussing about chromosomes. Chromosomes are the thread-like structures present inside the nuclear membrane. So these are the thread-like structures present inside the nuclear membrane. These chromosomes consist of DNA, RNA and histone proteins as well as non-histone proteins. Nothing but non-histone proteins, nothing but acidic proteins. These chromosomes constitute DNA, RNA. DNA means deoxyribose nucleic acid and RNA is ribose nucleic acid, and we have histone proteins are present, and also along with the histone proteins, non-histone proteins are also present, and these constitute the chromosomes. So chromosomes consist of DNA made up of DNA, RNA and also histone proteins as well as the non-histone proteins. The chromosomes are arranged in thread-like manner. We call them as chromatin. Chromosomes are arranged in thread-like structures. That is the substance of the chromosome. We called it as chromatin. The chromatin is of two types. One is uncoiled chromatin. One is highly uncoiled chromatin. Other one is highly coiled chromatin. Okay, the highly uncoiled chromatin we call it as euchromatin, and it is genetically active. Euchromatin is genetically active. And coming to the heterochromatin, it is genetically inactive. It is a highly coiled chromatin and it is genetically inactive. Okay, usually human body consists of 23 pairs of chromosomes. Usually human body consists of 23 pairs of chromosomes, means 46 chromosomes, right? Out of these 46 chromosomes, 22 pairs are autosomes. They help in the growth of the body. They help in the growth of the tissues of the body. 22 pairs of the autosomes, and one pair is the sex chromosome, which decides the sex character of the individual, which decides the sex of the individual. One pair of the sex chromosomes, otherwise called as gonosomes. Sex chromosomes, otherwise called as chromosomes. So totally we have 46 chromosomes. Out of these, 44 chromosomes are the autosomes, which helps in the growth of the body, and the remaining 2 chromosomes are the sex chromosomes that define the sex of the body. We know that in males we have XY chromosomes are present, and females is the XX. So in males we have XY and in females we have XX chromosomes are present. Okay, so this is about the how the chromosomes they decide the sex of the individual. Okay, and out of these 46 chromosomes, 23 pairs come from the mother. 23 chromosomes they come from the mother. 23 chromosomes come from the mother. They are maternal in origin. They come from the mother, and the remaining 23 chromosomes they come from the father. Nothing but they are paternal in origin. Paternal in origin. Thus, these chromosomes, that means we know that the gametes, the sex gametes are ova and sperm. They carry haploid number of chromosomes. So, when they unite, the resulting zygote consists of diploid number of chromosomes. So, out of these 46 chromosomes, 23 chromosomes come from the mother, that is maternal in origin, and 23 chromosomes come from the father, that is paternal in origin. Okay, then coming to the how the chromosomes divide. Okay, the chromosomes, what are the types of chromosomes? And the chromosomes, we know that these are the chromatids. These are the chromatids. We can see these chromatids like these chromatids only during the cell division, because during cell division what happens is the chromosomes are highly coiled and form thick rod-like structures. We call it as chromatids, which are united at the center by a centromere. Depending upon the position of centromere, we can divide the chromosomes into metacentric chromosomes, submetacentric chromosomes, satellite chromosomes and telocentric chromosomes. So what are metacentric chromosomes? Means the centromere is situated at the center, and these are the sister chromatids. They are of equal length. Sister chromatids are of equal length. Okay, the centromere is present at the center. We call it as submetacentric chromosomes. And here the centromere is present at a little higher middle rather than the middle region. It presents at a higher region. Okay, that's why the upper arms are comparatively shorter than the lower arm. The lower has longer arms, upper has shorter arms. And coming to the acrocentric chromosomes, the upper arms of the acrocentric chromosome, they show secondary constriction. So the area of the chromatid above the secondary constriction, we call it as satellite bodies. So we call them as acrocentric chromosomes. They consist of, the acrocentric chromosomes consist of satellite bodies. Okay. And coming to the telocentric chromosomes, they doesn't have any upper arms, only lower arms are present, and here the centromere is situated at the top. So when the centromere is situated at the center, we call them as metacentric chromosomes. When the centromere is situated a little above than its normal middle position, we call them as submetacentric chromosomes. When the centromere is present, and in the acrocentric chromosomes, centromere is situated similar to the submetacentric chromosome, but here a secondary constriction appears in the upper arms, and the centromere and the chromatid which is present above the upper arm, we call it as present above the secondary constriction, we call them as satellite bodies. And coming to the telocentric chromosomes, here the centromere is situated at the top, here the upper arms of the chromatid are absent. Okay, then coming to the how the genes are linearly arranged in the chromosomes. The genes are linearly arranged in the chromosomes. The genes are linearly arranged in the chromosomes. If two chromosomes, they exhibit the arrangement of genes are the same. If the chromosomes, they exhibit the genes arrangement is same, then we call them as homologous chromosomes. In homologous chromosomes, the arrangements of genes are same. Okay, so the position of a gene on the chromosome, we call them as locus. The position of a gene on the chromosome, we call it as locus. So in the homologous chromosomes, what happens here is, in the homologous chromosomes, the genes are arranged in the same manner. And in non-homologous chromosomes, what happens here is, for example, when we take XY chromosomes, here in the X, this is the X chromosome, and we take the Y chromosome. So the chromosome consists of both homologous and as well as non-homologous parts. And here the arrangement of genes is different. These are the non-homologous chromosomes. Non-homologous chromosomes. We can see in the XY pattern, non-homologous chromosomes. Okay. So depending upon the size and depending upon the position of centromere, the chromosomes are divided from A to G. The chromosomes are divided into A to G depending upon the position of the centromere as well as the size of chromosomes. The chromosomes are divided. We call this as karyotyping. So karyotyping helps in the observation of various syndromes like Turner syndrome, and also Down syndrome is present, Klinefelter syndrome is present. So karyotyping helps in useful for the diagnosing of various syndromes. This is nothing but the karyotyping is nothing but the arrangement of chromosome along with the size and as well as the position of centromere. So coming to the karyotyping. Coming to the karyotyping here, we have first group is the A group. In the A group, we have large chromosomes and they are metacentric chromosomes. Large group consists of A group consists of large chromosomes. The chromosomes are 3 pairs here, and they are metacentric chromosomes. They are situated at the center. And coming to the B group consists of 4 and 5 sets of chromosomes. Here they are submetacentric chromosomes because the centromere is situated at a higher level. Okay. And coming to the C group consists of 6 to 12 pairs of chromosomes. They are of medium size and they are also submetacentric. And coming to the D group, in the G group we will see acrocentric chromosomes. That means satellite bodies are seen here, right? Secondary constriction is seen in the upper arm, and the part of the chromatid which is present above the secondary constriction, we call it as satellite bodies. Okay. So this is the D group consists of acrocentric chromosomes of medium size, which consists of 3rd, 4th, 13, 14 and 15 pairs of chromosomes, right? And coming to the E group consists of 16, 17 and 18 pairs of chromosome. So it consists of submetacentric chromosomes which are of small size. It consists of submetacentric chromosomes which are of small size. And Grep group consists of 19 and 20 sets of chromosomes. They are metacentric chromosomes, are very small in size. And coming to the Z group consists of 21 and 22 chromosomes which are very small and acrocentric chromosomes, and here we have XY chromosomes is present in male. When we take female, another X chromosome is present, another X chromosome is present, nothing but submetacentric, another X chromosome present. So here we have karyotyping. It decides the sex of the body, sex of the human body. So if the XX chromosomes are present, then we have the individual is female. For example, if one more X chromosome is present here, one more X chromosome is present here, then the female is the super female, right? For example, here 2 X chromosomes are present and 1 Y chromosome is present. 2 X chromosomes are present and 1 Y chromosome is present. This condition we call it as Klinefelter syndrome. So the individual, of course, Y chromosome is present and the individual here is a male, but he exhibits some female characters also, like gynecomastia is seen, and we can be in Klinefelter syndrome individual, gynecomastia is seen, development of breast is seen, and also we can see the secondary sexual characters are not well developed. Smooth, that means mustache is not well developed in that individual, in that male individual, and also beard cannot be seen, and the external genital organs also underdeveloped in the Klinefelter syndrome individual. For example, if you take Turner syndrome, Turner syndrome is X0, right? In the Turner syndrome, X0 is present. That means only one chromosome is present. X chromosome is present, and then X or Y chromosome is absent. So in a Turner syndrome, only one X chromosome is present. The individual, of course, because of the X chromosome, the individual is female, but some characteristic features are present in the Turner syndrome, like webbed neck is present, low hair posterior leg is present, and also secondary sexual characters are not developed. So in that is the example of, that is the Turner syndrome. So because of this karyotyping, we will able to find out the various syndromes. Okay, that is the arrangement of chromosomes. So that's about the karyotyping. And one more important thing is barred body. So barred body, it is nothing but an inactive sex chromatin. In females, we have 2 X chromosomes are present. Out of these X chromosomes, 1 X chromosome, it is inactive. It is highly coiled. That inactive sex chromatin, we call it as barred body. So what does barred body means? It is inactive sex chromatin. Inactive sex chromatin is called barred body, and they are first founded by Barr and Bethram in the female cats. In the female cat, they are first, barred body is first identified by Barr and Bethram, 2 scientists, Barr and Bethram, in the female cats. So inactive sex chromatin is called barred body, and it is a plano-convex body situated near to deep to the, it is a plano-convex body situated deep to the nuclear membrane. When we examine, this is a nuclear membrane. It is a plano-convex body situated deep to the nucleus membrane. If the barred body is present, then it is having an inactive sex chromatin is present. Sometimes what happens here is 2 barred bodies are seen. Sometimes here 2 barred bodies are seen, then the female is a super female, because we can see the barred bodies is nothing but inactive sex chromatin. In males, barred body is absent because they have XY chromosomes, right? So out of this, one is active X chromosome and next one is Y chromosome. Females only we will able to see barred body. For example, in Klinefelter syndrome also, the genotype is the XSY, right? So in Klinefelter syndrome also, we will able to see the barred body. Okay, how can they determines the sex of the tissues. It determines the sex of the tissues. We can take the smear from the bucco-nasal region. We can take the smear from the bucco-nasal region here, and when we examine that, if we observe the body, then the individual, the syndrome of the individual or the sexual characters of the individual can be known. So that's about the barred body. Barred body is nothing but the inactive sex chromatin which is present in females and absent in males. During If two barred bodies are seen inside the nuclear membrane, then it is a superfemale. In males also, the barred bodies are present by in case of syndromes like Klinefelter syndrome. The phenotype is the XS5. In case also we can see the barred body. So that's about the chromosomes. So chromosomes are the thread-like structures. Okay, the chromosomes are the thread-like structures. The thread-like structures we call it as chromatin, and these chromosomes are made up of DNA, RNA, as well as histone or non-histone proteins. Usually we have 23 pairs of chromosomes are present. Out of this, one set comes from the mother and another set comes from the father, that is paternal and nodule. Together forms zygote consists of diploid number of chromosomes. So that's about the chromosomes.